a lot for the opportunity to present. Thanks for staying alive at 12 in the last day of the conference. Hope it will be interesting enough to listen. So I'm at the Max Planck in Tübingen, one of the meccas of Arabidopsis natural diversity. But I actually come from a microbiology background. So I did my PhD at the Weizmann Institute on bacteria, and more specifically on bacterial immunity, about how bacteria defend themselves against bacteriophages, against viral infection. And one of the cool things we discovered there was that some of the immune systems that we found in bacteria are actually things that we knew from eukaryotes. So my specific work was on T-domain proteins that were discovered as developmentally essential in Drosophila in Tübingen, where I work today, which is very exciting, were later discovered to be immune relevant in plants, were later discovered to be the backbone of animal innate immunity in toll-like receptors. And our contribution was to show that also already in bacteria, these proteins are immune-related, suggesting that many of the immune systems that we see in eukaryotes have their origin in prokaryotes. That was cool as an example, but it turned out to be a general principle, and we now know of over 10 such cases where the origins of eukaryotic immunity comes from bacterial immunity. And if I summarize the two things that I learned while working on this kind of project, was that first, the systems are conserved across the tree of life. So you could use these kind of homologies to study them in the different domains. And second, the way we discovered these systems in bacteria to begin with was not through homology searches, but through their genomic signatures. And this demonstrated the amazing power of being able to do homology-free work using genomic signatures to discover new components of immunity. And so it was very nice working with bacteria, and for some reason, I just like plants, so I want to work on plants. I do not recommend it as a model system, but I made a switch to plants, really because of a love of plants and botany. And for that, I came to Tübingen to try to see if we can implement the lessons that we learned from studying bacterial immunity in plant immunity. So how one can search for new immune system or immune genes in plant genomes. So the work that we do in my group basically covers the two extremes of evolutionary dynamics. On the one hand, we take advantage of the observation that immune systems can be conserved across the tree of life and ask, can we find new genes in plants that are homologous to bacterial immune systems but are not known to be involved in immunity in bacteria, and try to implicate them in immunity in this way. And the other thing is, can we use the diversity and the evolutionary dynamic signature of immune genes to identify genomic signatures in plant genomes that would allow us to discover new things? The first part first. So the first part started when we noticed that this bacterial immune gene that was very recently discovered, no one knows how it functions in bacteria, actually has a pretty good homologue in plants, including in Arabidopsis, which is nice. You can see that structurally, okay, I'll try forget about it. You can see that structurally it has three distinct domains, and these domains are very well aligned between the two systems. Now, in bacteria, we know that this thing is involved in phage defense. Again, we don't know the mechanism, but we know that if you're overexpressed in E. coli, it becomes resistant to certain phages. So the question was, is this homology also indicative of an immune function or viral interaction function in plants? So how can one test that? One is Eirik, is the PhD student who is bravely leading this project for the last 3 years. It wasn't easy. So the way we tested it was to go to the model plant of plant-virus interaction, Nicotiana benthamiana, an Australian plant, that is first amenable to infection with many viruses, and second, amenable to knockdown, VIGS-mediated knockdown that could allow us to test that. So the hypothesis is that if we knock down this gene in benthamiana, we would expect to see increased viral accumulation. And after 2 years of calibration, I'm happy to present this result that showed that indeed we were able to generate two independent silencing constructs that upon multiple replications were able to show us an increase in the two left parts of the slide. You can see an increase in the viral accumulation, and this is statistically significant. This increase is virus specific, so it is not general, which encourages us to think that it's not some general growth defect or some other general phenomenon that the knockdown is causing that might be generating this effect. My interest was in plants, but to tell the truth, this protein that we saw in plants is not just a plant protein. So first of all, we find it all across plants. We find it in bryophytes, we find it in monocots, and we find dicots, basically in every plant lineage. But we also find it in animals, which is nice because it means that maybe this observation can be expanded to other systems as well. And we even find it in vertebrates. Vertebrates, especially fish, tend to have an extended version where it has the conserved part, but an additional part that is unique to them. And we also find it in fungi, but in fungi, we find not the version that we find in plants, but a version that is much closer to what we see in bacteria. And that and the sporadic distribution across the fungal tree suggests that the origin of the fungal ones are actually later AGT and not the ancestral thing. We probably lost it in the opistocont separation. So that was a very cool discovery. We are really trying to wrap it up. And this is the first part. So I would say that this represents our first foray into asking, can we find new immune systems using conservation? I think we can. For the rest of my talk, I would like to talk about the diversity aspect, about how can we find genomic signatures of new immune systems that would lead us to things that are completely unknown, that are not homologous to things that we know. This signature, we expect it to be based on the evolutionary dynamic of immunity. And immunity is often described in the Red Queen race terminology. That means that both sides needs to evolve to keep in the same place. And the classical example for that in plant immunity are the most well-studied and biggest gene family of plant immune receptors called NLRs. They're also conserved or have homologs or similar proteins in animals. And the way it works is that these proteins have a leucine-rich repeat solenoid domain that is binding a bacterial component. When it does, there is an immune response and the pathogen cannot infect. So the bacterial protein or bacterial, viral, or pathogen protein keeps evolving to avoid this interaction, while the plant protein keeps evolving to maintain this interaction. And the way this evolutionary dynamics looks like on a plant genome is like this. So each row here represents a plant genome. They are all Arabidopsis. They are all Arabidopsis accessions, so it's all intraspecific diversity. And if you look at the locus that contain these genes, you can see that each accession of Arabidopsis contain a different complement of the light green genes, which are the NLR proteins. We are privileged to work in a plant that has an amazing natural diversity resource, Arabidopsis, through the work of Detlef Weigel, my advisor, and additional labs, Magnus Nordberg mainly. An amazing collection of the natural diversity of Arabidopsis has been generated. So we have collected accessions from across the distribution range of this species, and more importantly for me, we have really unparalleled genomic resources. So we have over 450 fully assembled, including the telomeres, including the centromeres, including everything, different natural accessions of this plant, and we also have the plants that were sequenced to play with. So using this amazing resource, basically what we're doing is we're looking for things that look like this, but are not NLRs, because I want to find new things. And I would like to share with you briefly some of the things that we found. So this is an example of the things that we really like to find. Each row, again, represents a different Arabidopsis accession. So these are all the same species. This is the same locus in their genome. And in the brightly pink, purple colors that my PhD student chose, you can see the variable genes. So you can see that in general, the genes to the left and genes to the right pretty much look the same, but these small genes, small colorful genes in the middle, seem to be pretty unique to every accession. What are these genes? These genes are genes from the defensin antimicrobial peptide family. So they belong to a family that some 4 members in Arabidopsis were described to be involved in antifungal immunity, but the Arabidopsis genome contains over 200 of them, and none of these ones that are viable have ever been shown to be involved in immunity, and I think this is, for example, a good source of new components that are involved in immunity. This is the work that Naya Ferber that is cloning them and throwing them on fungi, I think, as we speak. So I'm waiting for the email. But this was something that, you know, is kind of related to immunity. It has defensin in its name. Maybe it's not new enough. Let's look at this locus. This locus contains NLR genes, so known immune receptors in red. And as you can see, between the different accessions, the red ones are really different. Each accession has a different thing. But it's not just the reds, right? There are also like the blues look kind of diverse. What are the blues? The blues are a gene family that we call the double doof. Those who are not into protein domains, doof is an acronym for a domain of unknown function. And these proteins have 2 of them. We really don't know what they do. There's no paper about their function in any plant, but they are highly diverse in Arabidopsis. So this is like one of the candidates that we really want to get into. If anyone is into ER proteins, they are ER proteins, so I'm happy to chat. And there are other families. This was discovered by Anna during a master thesis. But the last point that I want to tell you about is what happens when you take this to extreme. So, you know, this is like a diversity that is reasonable to look at. You can understand kind of what is going on. When looking for generally diverse loci, we found these things. Now, you see the blue genes. So the blue genes in this locus can reach up to 95 copies. 95 copies of the same gene. These clusters exist only in a subset of the accessions, and we thought that this must be the most, you know, crazy thing ever. Turns out to be a proline synthesis enzyme, the most housekeeping normal metabolic gene that you could imagine. So we started to ask what is going on. And what is going on is that this enzyme in the reference genome and in most of the Arabidopsis accession is actually a single copy essential enzyme that live happily on its location on chromosome 5 of the Arabidopsis, but in some accession, for some reason, is translocated and generates these crazy duplication clusters on the centromere. When we looked at it a bit more, we saw that these clusters are there, but are completely silenced. They're methylated, except for the first copy. The first copy seemed to be expressed. What does it mean? We took an accession that has only the normal copy, only the ARM copy, where we saw that all the reads come from the ARM copy. This is a good control. There is a SNP that allows us to differentiate the 2 copies. We took accessions that have the duplication cluster and realized that this enzyme transcript pool actually comes from these 2 loci together. And then we found an accession where the entire enzyme transcript pool comes only from the extreme duplication cluster. This enzyme is an essential enzyme, which means that this plant now became dependent on the crazy duplication event. That was weird, but what was weird there is that we found 2 cases of that, where we have these translocation, extreme duplication, and silencing of the original copy, making the plants dependent on these extreme duplication events, making us think that this is a case where an enzyme, an essential enzyme, starts behaving as if it is a selfish element, utilizing its own essentiality for that. And the cherry on the cake is that all of this is happening in centromeric regions that are not supposed to be transcriptionally active at all, according to the textbooks, but this is the case. All this story is not related at all to immunity, I think, but it's still interesting and kind of shows you where you can get when you have really good genomes. This is a work of multiple students, Sandra who made the observation, Lucas who did the experimental, and Siching who is finalizing the bioinformatic analysis. So to sum it up, I hope I could show you that when you have really good genomes and when you come with, let's say, an outsider approach, you try to bring concepts from one field to another, it leads to interesting observations. The things that we're currently working on are these cases of ancient immunity, these weird cases of this selfish duplication, which became a thing. I did not think that I would work on any of that, but no. And what we were most excited about are the possibilities to find new immune genes in some of these interesting genomic loci that I briefly shared with you. And with that, I'd like to finish on time, and happy to take questions.